This is page 52 and we have got a synapse, and ACH or acetylcholine is the transmitter. It's drawn upside down to the one that you normally see. We usually do it at the presynaptic neuron at the top, but it'll work in the same way. Describe the part played by acetylcholine, 3 marks. So what have we got going here? We've got, this is a presynaptic neuron, so presynaptic neuron is here, and we've got our postsynaptic neuron up here, and if I do in green where our ACH is, acetylcholine, be in these vesicles here. And so what's going to happen is these vesicles are going to fuse with the presynaptic membrane, so they'll move and fuse with the presynaptic membrane, release their neurotransmitter into the synaptic cleft, and those are going to bind to receptors on the postsynaptic membrane, and that will then allow sodium to go in and cause a depolarization. So we just need to say 3 sensible things there. So acetylcholine is the neurotransmitter, so it's a neurotransmitter, and it will be released by exocytosis into the synaptic cleft, released by exocytosis into the synaptic cleft. It diffuses across the synaptic cleft. It will bind to these receptors on the postsynaptic membrane, bind to receptors on the postsynaptic membrane, and that will allow sodium channels to open and a depolarization, so that this depolarization can spread as an action potential down the neuron. So allows sodium channels to open, then we will get a depolarization or action potential. And hopefully those 3 marks are in there somewhere. Describe the function of the mitochondria shown. So mitochondria, and hopefully by now you're getting well used to saying what the mitochondria do. So I'd always just start off by saying what the role of mitochondria is. So mitochondria produce ATP, energy in aerobic respiration. Always a good place to start that. And these mitochondria are going to provide energy for the production of acetylcholine, movement of things within the cell, like exocytosis, active transport of the calcium out of the presynaptic neuron. So a few suggestions there. So for synthesis of acetylcholine, we could say pump, that's it for 1 mark, that's all you need to say, but I'll give you another suggestion. Pumping out of calcium ions as another suggestion there.